My dear students, welcome back to our channel. In this video, I will be explaining the B.Sc
first semester
subject Zoology, Animal Diversity as prescribed by Osmania University and other universities of Telangana. We will go through the important questions and answers one by one. Let's begin with a quick revision. Unit 1 first important short question is: define multicellularity. Answer: multicellularity means the condition where an organism is made up of many cells that work together to perform different functions. In simple words, it means many cells living and working as one body. In unicellular organisms like amoeba or paramecium, a single cell does all the work, feeding, moving, and reproducing. But in multicellular organisms like hydra, earthworm, or humans, there are many specialized cells. Each type does a specific job. For example, in hydra, some cells help in digestion, some form tentacles for catching food, and others help in reproduction. Multicellularity allows division of labor, meaning each cell type performs a unique function for the survival of the whole organism. It also helps in growth, repair, and complex body organization. In short, multicellularity is equal to many cells plus teamwork plus specialization, efficient and organized life. Next important short question is: to distinguish diploblastic and triploblastic organization. Answer: animals are grouped based on the number of cell layers formed during early development. These layers are called germ layers, which later form different tissues and organs. 1. Diploblastic animals. They have two germ layers, ectoderm, outer layer, and endoderm, inner layer. There is no middle layer, mesoderm. The body is simple, without complex organs. Example: hydra and jellyfish, phylum Cnidaria. 2. Triploblastic animals. They have three germ layers, ectoderm, mesoderm, and endoderm. The mesoderm gives rise to muscles, organs, and blood vessels. These animals show better body organization and organ systems. Example: planaria, phylum Platyhelminths, and all higher animals. Easy to remember. D is equal to 2 layers, simple animals. Tri is equal to 3 layers, advanced animals. So, the appearance of the mesoderm marks the evolution of complex body structures. Next important short question is: 3. Name 2 types of body symmetry in animals. Answer: body symmetry means the arrangement of body parts around a central line or point. It shows how an animal's body can be divided into equal halves. There are mainly 2 types of symmetry found in animals. 1. Radial symmetry. The body can be divided into similar halves through many planes passing through the center. These animals do not have a definite right or left side. Example: hydra, jellyfish, and starfish. Radial symmetry helps such animals interact with the environment from all directions, which is useful for sessile or slow-moving forms. 2. Bilateral symmetry. The body can be divided into 2 equal halves, left and right, by only one plane. It allows streamlined movement and development of a head region, cephalization. Example: planaria, earthworm, and humans, asterisk. Easy trick. Radial is equal to round like a V. Bilateral is equal to balanced left and right. Next important short question is: 4. Give the 3 types of body cavities, terms only. Answer: a body cavity, also called column, is the space between the body wall and the internal organs. It helps in free movement, growth, and proper functioning of organs. Based on the presence or absence of this cavity, animals are divided into three types. 1. Acoelomate. Animals without a body cavity between body wall and gut. Example, Planaria, phylum Platyhelminths. 2. Pseudocoelomate. Animals having a false body cavity, not completely lined by mesoderm. Example, Ascaris, phylum Nemathelminths. 3. Coelomate, Eucoelomate. Animals with a true body cavity completely lined by mesoderm. Example, earthworm, mollusks, humans. Easy to remember. A is equal to absent, pseudo is equal to false, coelomate is equal to true cavity. This classification shows how animals evolved from simple, no cavity, to complex, true column forms. Next important short question is 5. What is meant by parasitic adaptation? One example from helminths. Answer. Parasitic adaptation means the special features or changes developed by parasites to live successfully inside the host's body without being destroyed. These adaptations help the parasite to absorb food, reproduce, and protect itself from the host's immune system. In helminths, parasitic worms, these adaptations can be structural, physiological, or reproductive. Example. In Taenia solium, the pork tapeworm, several parasitic adaptations are seen. It has hooks and suckers on its head, scolex, to attach firmly to the intestine of the host. The body is flat to absorb digested food directly through the skin. It produces a large number of eggs to increase chances of survival. Easy tip to remember. Grip, absorb, multiply, attach, feed, and reproduce. These three are the main parasitic adaptations in helminths. Next, Unit 1, Invertebrates, Protozoa Platyhelminths. Long questions. First important long question is: describe general characters and classification of protozoa up to classes with representative examples. Answer. Introduction. Protozoa are the simplest and most primitive animals belonging to the kingdom Protista. They are unicellular, but each cell performs all vital life functions like movement, feeding, respiration, and reproduction. General characters of protozoa. 1. Unicellular and microscopic. The body is made of a single cell, but it performs all activities of a complete organism. Example, amoeba, paramecium. 2. Habitat. Most live in fresh water, marine water, or moist soil. Some are parasitic, living inside host body. 3. Body structure. Body is surrounded by a thin plasma membrane. In some, a pellicle gives shape and protection. 4. Locomotion. Movement is by pseudopodia, flagella, or cilia. 5. Nutrition. May be autotrophic, heterotrophic, saprozoic, or parasitic. 6. Reproduction. Usually by binary fission, asexual, sometimes sexual reproduction, syngamy or conjugation, occurs. 7. Symmetry. Usually asymmetrical or radial in some forms. 8. Respiration and excretion. Through diffusion over the body surface. Classification of protozoa up to classes. According to locomotory organs and mode of life, protozoa are classified into 4 main classes. 1. Class Rhizopoda, Sarcodina. Locomotion by pseudopodia. Example, Amoeba proteus, Entomoeba histolytica. 2. Class Mastigophora, Flagellata. Move with flagella. Some are free-living, others parasitic. Example, Euglena, Trypanosoma. 3. Class Ciliophora, Ciliata. Have cilia for movement and feeding. Complex body with 2 nuclei. Example, Paramecium, Vorticella. 4. Class Sporozoa. No locomotory organelles in adult stage. All are parasitic, reproducing by spores. Example, Plasmodium causes malaria. Conclusion. Protozoa show how life evolved from simple to complex forms. Though microscopic, they play a big role in nature. Some are beneficial in food chains, while others cause diseases. Easy tip: amoeba crawls, euglena swims, paramecium dances, plasmodium hides. Next important long question is: to explain the locomotion mechanisms in protozoa, illustrating major locomotory organelles and their functional roles. Answer. Introduction. Locomotion means movement from one place to another. In protozoa, locomotion is very important for searching food, escaping from enemies, and finding suitable conditions. These organisms move with the help of special structures called locomotory organelles. The main types of locomotory organelles in protozoa are pseudopodia, flagella, and cilia, each with its own movement mechanism. 1. Pseudopodia, false feet. Found in amoeba, class Rhizopoda. These are temporary finger-like projections of the cytoplasm. Movement occurs by the flow of cytoplasm, amoeboid movement. The cell pushes its cytoplasm forward to form a pseudopodium, then the rest of the body flows into it. Function, also used for capturing food by phagocytosis. Example, amoeba proteus. 2. Flagella. Found in flagellates like euglena and trypanosoma. Flagella are long, whip-like structures made of microtubules. Movement occurs when flagella beat in a wave-like manner, pulling or pushing the organism through water. Function, used for both locomotion and sometimes feeding. Example, euglena swims smoothly using its single flagellum. 3. Cilia. Found in ciliates like paramecium. Cilia are short, hair-like structures present all over the body surface. They beat in a coordinated rhythm, producing rapid movement through water, like oars of a boat. Function, help in locomotion and also in directing food towards the mouth, cytostome. Example, paramecium moves swiftly with ciliary action. Conclusion. Different protozoans have evolved different locomotory organelles suited to their lifestyle. Amoeba crawls with pseudopodia. Euglena swims with flagellum and paramecium glides with cilia. Easy to remember. Pseudopodia crawling, flagella swimming, cilia gliding. 3 styles, 1 goal: movement for life. Next important long question is: 3. Describe porifera, general characters, classification up to classes, canal systems, types, and the structure function of spicules. Discuss evolutionary significance of sponges as early metazoans. Answer. Introduction. Porifera, commonly called sponges, are the simplest multicellular animals. They are mostly marine, sessile, and live attached to rocks or other substrates. Despite their simple body, they show cellular level of organization, meaning cells are specialized but not organized into tissues or organs. General characters of porifera. 1. Body structure, porous and perforated by tiny holes called ostia. The main body cavity is spongosoil, opening to the outside via osculum. 2. Symmetry, usually asymmetrical or radial. 3. Skeleton, made of spicules, calcium carbonate or silica, or spongin fibers. 4. Nutrition, filter feeders, water-carrying food particles flows through canals. 5. Reproduction, both sexual and asexual, budding, gemmules. Classification of porifera up to classes. 1. Class Calcarea, calcium carbonate spicules, small, marine. Example, Cycon. 2. Class Hexactinellida, siliceous spicules, 6-rayed, deep-sea sponges. Example, Euplectella. 3. Class Demospongi, siliceous spicules or spongin, largest group, marine and freshwater. Example, Spongilla. 4. Class Homoscleromorpha, simple siliceous spicules, marine. Example, Oscarella. Canal systems, types. 1. Asconoid, simplest, small, tubular, example, Leucosolenia. 2. Syconoid, folded canals, moderate size, example, Sypha. 3. Leuconoid, highly branched canals, large and complex, example, Spongilla euspongia. Spicules, structure and function. Spicules are needle-like structures of calcium carbonate or silica. Function, provide support, protection, and maintain the body shape. Evolutionary significance. Represent the transition from unicellular to multicellular animals. Show cellular specialization without tissues. Early sponges helped in understanding evolution of metazoans. Easy to remember. Porifera is equal to pores plus filter feeders plus spicules. Asconoid is equal to simple tube. Syconoid is equal to folded tube. Leuconoid is equal to branched tube. Sponges are living fossils, showing how life became multicellular and laid the foundation for complex animals. Next important long question is: 4. Describe cnidaria, general characters, and classification up to classes. Explain polymorphism in hydrozoa and siphonophora and the ecological significance of coral reef formation. Answer. Introduction. Cnidaria is a phylum of radially symmetrical, diploblastic animals that are mostly aquatic and carnivorous. They are soft-bodied and exhibit tissue-level organization with stinging cells, cnidocytes, for capturing prey. General characters of cnidaria. 1. Body structure, diploblastic, ectoderm plus endoderm, with mesoglia in between. 2. Symmetry, radial symmetry. 3. Body forms, 2 main forms. Polyp, sessile, tubular, example, hydra. Medusa, free-swimming, umbrella-shaped, example, Aurelia. 4. Digestive system, incomplete, with a gastrovascular cavity. 5. Reproduction, both sexual, medusa, and asexual, polyp. 6. Locomotion, polyp is sessile, medusa swims with bell contraction. 7. Cnidocytes, specialized cells with nematocysts for defense and prey capture. Classification up to classes. 1. Class Hydrozoa, mostly marine, both polyp and medusa present. Example, Obelia. 2. Class Scyphozoa, true jellyfish, medusa dominant. Example, Aurelia. 3. Class Cubozoa, box jellyfish, strong swimmers, potent sting. Example, Chironix. 4. Class Anthozoa, only polyps, corals and sea anemones. Example, coral, Actinia. 5. Class Staurozoa, stalked jellyfish, attached to substrates. Example, Stauromedusa. Polymorphism in hydrozoa and siphonophora. Polymorphism is equal to presence of different types of individuals in a colony performing specialized functions. In Obelia, gastrozooids feeding, gonozooids reproduction, dactylozooids defense. In Physalia, Portuguese man-of-war, siphonophora, polyps are specialized for flotation, feeding, reproduction, and defense. Ecological significance of coral reefs. 1. Habitat, provide shelter and breeding grounds for fish and marine life. 2. Protection, protect coasts from waves and erosion. 3. Biodiversity, support rich marine biodiversity. 4. Economic importance, source of food, medicine, and tourism. Easy tip to remember. Cnidaria is equal to stinging plus 2 body forms plus polymorphic colonies plus coral reefs protect life. This phylum shows how tissue-level organization and division of labor evolved in early animals. Next important long question is: 5. Describe platyhelminths and nemathelminths, general characters and classification up to classes, and compare key parasitic adaptations in helminths. Answer. Introduction. Helminths are worm-like invertebrates divided mainly into platyhelminths, flatworms, and nemathelminths, roundworms. Many are parasitic, living inside other animals or humans, while some are free-living. General characters. 1. Platyhelminths, flatworms. Body flattened dorsoventrally, soft, unsegmented. Triploblastic and acoelomate, no body cavity. Bilateral symmetry with cephalization, head region. Digestive system, incomplete, single opening. Nervous system, ladder-like with ganglia and nerve cords. Example, planaria, free-living, tania, parasitic. 2. Nemathelminths, roundworms. Body cylindrical, elongated, unsegmented. Triploblastic, pseudocoelomate, bilaterally symmetrical. Complete alimentary canal with mouth and anus. Nervous system, simple, with nerve ring. Example, Ascaris, Buchereria, parasitic. Classification up to classes. Platyhelminths. 1. Turbellaria, mostly free-living. Example, planaria. 2. Trematoda, flukes, parasitic. Example, fasciola. 3. Cestoda, tapeworms, parasitic, segmented. Example, tania. Nemathelminths, nematoda. 1. Adenophoria, enoplia, mostly free-living. Example, trichinella. 2. Cesarnenti, chromadoria, many parasitic. Example, Ascaris, Buchereria. Key parasitic adaptations in helminths. Feature function example. Attachment organs hooks, suckers to stay in host tania. Protective covering thick cuticle or tegument resists host digestion fasciola. High reproductive capacity produce thousands of eggs to ensure survival Ascaris, tania. Reduced digestive system absorbs food directly from host tapeworms. Life cycle adaptations multiple hosts, resistant larval stages fasciola, Buchereria. Conclusion. Platyhelminths and nemathelminths show how worms adapted to parasitic life, ensuring survival inside hosts. Easy tip to remember. Flatworms is equal to flat plus attach plus multiply. Roundworms is equal to cylindrical plus complete gut plus survive. These helminths are important in medicine and ecology, as some cause human and animal diseases. Next important long question is 6. Explain regeneration in turbellarians, patterns, mechanisms and experimental evidence. Answer. Introduction. Regeneration is the ability of an organism to regrow lost or damaged body parts. Turbellarians, a class of free-living flatworms, phylum Platyhelminths, are famous for their high regenerative power. Patterns of regeneration. 1. Anterior regeneration. When the tail is cut off, it regrows a new head. Example, planaria regenerates its head after decapitation. 2. Posterior regeneration. When the head is cut off, a new tail is formed. 3. Lateral or fragment regeneration. If the body is cut transversely or into small pieces, each fragment can regenerate into a complete worm. Example, cutting a planaria into 3 parts forms 3 complete worms. Mechanism of regeneration. 1. Wound healing. Immediately after injury, the wound closes to prevent infection. 2. Cellular response. Specialized stem cells called neoblasts migrate to the wound site. Neoblasts divide and differentiate into various cell types. 3. Blastema formation. Accumulation of undifferentiated cells at the cut site forms a blastema, which develops into the missing parts. 4. Polarity determination. The worm recognizes which end is head and which is tail using chemical signals, ensuring proper regrowth. Experimental evidence. Cutting experiment. Planaria is cut transversely or longitudinally. Observed that each piece regenerates into a complete worm, proving totipotency of neoblasts. Chemical treatment. If cell division is blocked, regeneration fails, showing that cell proliferation is essential. Conclusion. Regeneration in turbellarians demonstrates amazing plasticity of cells and provides a model to study stem cell biology. Easy tip to remember. Cut it, heal it, grow it, complete it. Tail regrows head. Head regrows tail. Body fragment new worm. Turbellarians are thus natural experts of regeneration, making them important for biology and medical research.